Hello everyone, welcome to my channel. Today the topic of discussion is retinal anatomy with respect to OCT cross section. I have no financial interest with this video. This is solely the retina is derived from the Latin word rete. Rete in Latin means a net. Now in the most comprehensive sense, retina includes all the structures that are derived from the optic vesicle, namely the sensory layers of the retina, which is the pars optica retinae, the pigmented epithelium of the retina, as well as the epithelial linings of the ciliary body, which is the pars retinae ciliaris, and of the iris, which is the pars retinae iridis. However, the retina can be grossly divided into two layers: the sensory layer, which is derived from the inner layers of the optic vesicle, and the pigmented layer, which is derived from the outer layer of the optic vesicle. Now this is a schematic drawing of the part of sensory retina attached to the retinal pigment epithelium and the Bruch's membrane. The sensory and the pigmented layers are attached loosely to each other by the extracellular matrix that fills the space between the apical villi of the pigment epithelium and the outer segments of the photoreceptors, as well as the interphotoreceptor space. Looking into the topography of the retina proper is a thin, delicate layer of nervous tissue that has a surface area of about 266 mm square. Now the major landmarks of the retina are the optic disc, the retinal blood vessels, the area centralis with the fovea and the foveola, the peripheral retina, which includes the equator and the ora serrata. The retina is thickest near the optic disc, where it measures 0.56 mm. It becomes thinner towards the periphery, the thickness reducing to 0.18 mm at the equator and to 0.1 mm at the ora serrata. The retina is divided into two regions: the central retina, or the area centralis, or the posterior pole, which includes the optic disc and the macula lutea, and the peripheral retina. Now the macula lutea is a region which consists of the umbo, foveola. These two regions makes up the foveal avascular zone, the parafovea and the perifovea. Now since we are understanding the anatomy of the retina based on the OCT, so we need to understand what does an OCT do. Well, OCT performs high resolution cross-sectional tomographic imaging of the internal microstructure of the retina by measuring back reflecting and back scattering light waves from different microstructural features within the material or tissue. So therefore, in an OCT image, the fiber layers and the layer boundaries which strongly reflect light are highly reflective, and the cellular layers which weakly reflect light are weakly reflective. So therefore, firstly, the cellular layers would appear as weakly reflective layers. The fibrous layers and the boundaries will appear highly reflective layers. Now the OCT scan of the retina represents its 10 different layers. Now since the retina that we see clinically is from the vitreous, and thus we have the internal limiting membrane end face to our eyes, we will be discussing the layers in that order. That is, from the inner retina to the outer retina. So firstly, we see a long dark gap before the retinal layers, which is the vitreous, and it is a hyporeflective layer. Then we have the internal limiting membrane, which is an intermittently hyporeflective layer just at the junction of the vitreous and the retina. Second layer that we see is a fibrous nerve fiber layer, which is a hyperreflective layer. The third layer that we see is a cellular layer, the ganglion cell layer, which is a hyporeflective layer. Fourth layer is the hyperreflective inner plexiform layer. Fifth layer is the hyporeflective inner nuclear layer. The sixth layer is the hyperreflective outer plexiform layer. The seventh layer is the hyporeflective outer nuclear layer. The eighth layer is the hyperreflective boundary, that is the external limiting membrane. Ninth layer is the layer of rods and cones, or the photoreceptor layer, which is first a hyporeflective band, followed by a hyperreflective band, and again followed by a hyporeflective band. The 10th layer is the hyperreflective retinal pigment epithelium. Now, there are exceptions that occur in this image. Firstly, the Henle's fiber layer is a fibrous layer, yet it is a hyporeflective layer. Now, the reason is that the nerve fibers of Henle's fiber layer tilt forward and towards the periphery of the macula. The measurement beam enters at normal retina in a significant angle to the fiber's orientation rather than entering it in a parallel way. Thus, Henle's fiber layer becomes weakly reflective and becomes indistinguishable from the similarly weakly reflective outer nuclear layer. However, when OCT measurement beam enters the pupil from an eccentric position, the beam obliquely reaches the retinal surface and consequently passes the retina perpendicular to the Henle's fiber layer on one side. This increases the reflectivity of the Henle's fiber layer, causing it to be a highly reflective layer. This is similar to when the retinal layer tilts as a result of retinal detachment or drusen. The axonal endings of the synapse phase of the outer plexiform layer combine with tight junctions to form a sheet-like structure acting as the boundary surface with the highly reflectivity. And another exception is the retinal pigment epithelium, which is a cellular layer, yet it is a hyperreflective layer. Now, the reflectivity of the RP is thought to be due to melanin pigment. However, the RP can be seen as two distinct lines in 3 micrometer ultra high resolution OCT. Now, the anatomy of the fovea is different from the anatomy of the rest of the retina. Fovea is a very specialized structure in the retina which gives us the best visual acuity. The structures included in the fovea are the retinal pigment epithelium, the photoreceptor layer, where only cone cells are present, the external limiting membrane, the outer nuclear layer, which contains the nuclei of the cone cells, the inner fibers of the photoreceptors, the so-called Henle's fiber layer, and the internal limiting membrane. Now, since retina is a specialized nervous layer, it contains 3 different types of neurons with dedicated functions to it. The primary neurons in the visual pathway are the photoreceptors, which with their end organs, pericardia, axonal processes, and synapses constitute the layer of rods and cones, the outer nuclear layer and the outer plexiform layer. The job of these layers is to perceive the information in its form. The bipolar, horizontal, amacrine, and interplexiform cells constitute the second order neurons. The ganglion cell layer and the nerve fiber layer form the conductive network internal to the inner plexiform layer and external to the internal limiting membrane. Thus, they contribute in conducting the received impulse to the adjacent layers and also contributes in association between the third order and the first order neurons. This network of third order neurons in the ganglion cell layer and the nerve fiber layer and their respective axonal processes conduct the information gathered by the photoreceptors for further processing to the visual cortex. The Mueller cells extend between the 2 limiting membranes and together with other glial cells constitute the neuronal connective tissue cells of the retina. So friends, this is the part 1 of our retinal anatomy. In the second part, we will be discussing the retinal layers in more details. Please like, subscribe and share the video. Thank you.